Human Physiology, Sodium Balance. Sodium is an important cation distributed primarily outside the cell. The cell's sodium concentration is about 15 millimolar per liter, but this varies in different organs, and with an intracellular volume of 30 liters, about 400 millimolar are inside the cell. The plasma and interstitial sodium is about 140 millimolar per liter, with an extracellular volume of about 13 liters. 1800 millimolar are in the extracellular space. The total body sodium, however, is about 3700 millimolar, as there is about 1500 millimolar stored in bones. The usual sodium intake of an Australian diet is about 180 millimolar per day, but varies widely, between 50 and 400 millimolar per day, depending on habit and cultural influences. The body has potent sodium-retaining mechanisms, and even if a person is on 5 millimolar of sodium a day, they can maintain sodium balance. Extra sodium is lost from the body by reducing the activity of the renin-angiotensin-aldosterone system, which leads to increased sodium loss from the body. Sodium is lost through the kidneys, sweat, and feces. In states of sodium depletion, aldosterone levels increase, and in states of sodium excess, aldosterone levels decrease. The major physiological controller of aldosterone secretion is the plasma angiotensin II level, which increases aldosterone secretion. A high plasma potassium also increases aldosterone secretion, because besides retaining sodium, high plasma aldosterone causes potassium loss by the kidney. Plasma sodium levels have little effect on aldosterone secretion. A low renal perfusion pressure stimulates the release of renin, which forms angiotensin I, which is then converted to angiotensin II. Angiotensin II will correct the low perfusion pressure by causing constriction of blood vessels and by increasing sodium retention by a direct effect on the proximal renal tubule and by an effect operated through aldosterone. The perfusion pressure to the adrenal gland has little direct effect on aldosterone secretion, and the low blood pressure operates to control aldosterone via the renin-angiotensin system. In addition to aldosterone and angiotensin II, other factors influence sodium excretion. Thus, in high sodium states due either to excess intake or cardiac disease, atrial peptide is secreted from the heart and by a series of actions causes loss of sodium by the kidney. Elevated blood pressure will also tend to cause sodium loss, and a low blood pressure usually leads to sodium retention. Aldosterone also acts on the sweat ducts and colonic epithelium to conserve sodium. When aldosterone has been activated to retain sodium, the plasma sodium tends to rise. This immediately causes release of ADH, which causes water to be retained, thus retaining sodium and water in the right proportion to restore plasma volume. Potassium balance. Potassium is predominantly an intracellular ion, and most of the total body potassium of about 4,000 millimolar is inside the cells, and the next largest proportion, 300 to 500 millimolar, is in the bones. Cell potassium concentration is about 150 millimolar per liter, but varies in different organs. Extracellular potassium is about 4 millimolar per liter, and with an extracellular volume of about 13 liters, 52 millimolar, less than 1.5%, is present here, and only 12 millimolar in the plasma. In an unprocessed diet, potassium is much more plentiful than sodium and is present as an organic salt, while sodium is added as NaCl. In a hunter-gatherer, potassium intake may be as much as 400 millimolar per day, while in the Western diet it is 70 millimolar per day, or less if a person has a minimal amount of fresh fruit and vegetables. Processing of foods replaces potassium with sodium chloride. While the body can excrete a large potassium load, it is unable to conserve potassium. On a zero potassium intake, or in a person with potassium depletion, there will still be a loss of potassium and 30 to 50 millimolar per day in the urine and feces. If there is a high potassium intake, for example, 100 millimolar, this would potentially increase the extracellular potassium level 2 times before the kidney could excrete the extra potassium. The body buffers the extra potassium by equilibrating it within the cells. The acid-base status controls the distribution between plasma and cells. A high pH, alkalosis, the pH greater than 7.4, favors movement of potassium into the cells, while a low pH, or acidosis, causes movement out of the cell. A high plasma potassium increases aldosterone secretion, and this increases the potassium loss from the body, restoring balance. This change of distribution with the acid-base status means that the plasma potassium may not reflect the total body content. Thus, a person with an acidosis and a plasma potassium of 6.5 millimolar per liter could be depleted of total body potassium. This occurs in diabetic acidosis. Conversely, a person who is alkalotic with a plasma potassium of 3.4 millimolar per liter may have normal total body potassium.